days. Thank you so much, sir, for honoring our invitation. The floor is yours.
Thank you very much, Abdul Basit and Musab, for the invitation and to give me this opportunity to exchange with the PhD student, master student, postdoc, and some professors if they are connected. And it's always a pleasure to participate in the scientific activities for Africa. I'm originally from Morocco. I'm African, and it's very, very important to push and to encourage people in Africa to develop research, because we need research everywhere, and it's very, very important. And I really wish to thank you. And today we talk about the contribution of animal models in understanding and treating Parkinson's disease. And the animal models are very, very important in advancement of scientific knowledge. And we need to understand the physiological and the pathological mechanisms of normal functions, and at the same time of pathologies, human pathologies. And without animal models, we cannot understand very well these pathological mechanisms. And the consequence will be that we cannot develop new therapeutic approaches for human diseases. At this time, more than ever, we realize the importance of biomedical scientific research because of this pandemic, the SARS-CoV-2 coronavirus pandemics. And the fight against this coronavirus requires research on animal experiments. And if you look to the vaccines that they are developed everywhere in the world now, you can see that all vaccines currently in clinical trials were first tested in animals. And this is a demonstration that without animal models, without animal experiments, we cannot develop vaccines and we cannot fight against the SARS-CoV-2. Now, everyone is asking, are we allowed to experiment on animals? And according to Claude Bernard, who is a scientific medical doctor, French medical doctor, in his introduction to the study of experimental medicine that he published in 1865, he said that, as for me, I think that we have this right in a complete and absolute way. It would be very strange, in fact, to recognize that man has the right to use animals for all purposes of life, for his domestic services, for his food, and to forbid him to use them to educate himself in one of the sciences most useful to mankind. Therefore, if it is immoral to make an experiment on a man as soon as it is dangerous for him, although the results may be useful to others, it is essentially moral to make experiments on an animal, although painful and dangerous for him, as soon as they may be useful for man. And he was really very, very interested in experimental medicine that be necessary to use animal experiments. And I think these things that he said are also correct at our period of time. And if we look now to the animal experiment in the ethics, we can know that animal experiment is subject to very strict ethical regulations. Now we pay really attention to try to have a good use and strict ethical use of animals in biomedical research. And animal experiment is submitted to a law, and there is in Europe a decree of October 19, 1987, which was revised in 2001. There is a very strict European directive of 2010, which was transposed into French law in February 2013. And in French now, we are really very, very strict on the use of animals in biomedical research. And in all the countries in Europe and United States and Canada, everywhere now, the researchers are very, very strict on the use of animal experiment. And animal experiment is subject to the rule of the 3R, which are replacement, reduction, and refinement. If we look to the replacement, we have to replace animal experiment when it is possible. Unfortunately, it's not always possible to replace with cell cultures or with modeling or your computer models. We have also to respect the reduce of the number of animals that we can use for an experiment. And for that, we have to standardize all the protocols. We have to pay attention to the implementation of statistics. We have to use the correct number of animals in an experiment to have good statistics to determine with the scientifically available results. And at the same time, we have to look to the evolution of the means of investigation, because all of this will help us to reduce the number of animals in every experiment. Sorry. Then we have to refine. We have to refine the protocols to minimize animal stress and the pain. And this have a very good result, which is the importance of animal welfare. What is a definition of animal model? Animal model is a non-human animal with a condition similar to a human disease and serves as a model for the study of this condition. And according to the American National Research Council Committee on Animal Models for Research and Aging, in biomedical research, an animal model is a model in which it is possible to study a spontaneous or induced pathological process that has one or more aspects in common with an equivalent phenomenon in human. And this is very, very important, because without the definition, we cannot know exactly how we can use an animal model. And in the case of the study of human disease, there are 3 criteria. We have the phase validity, the construct validity, and the predictive validity. And for the first one, for the phase validity, the symptoms observed in animals should be similar to those observed in humans. If you don't have similar symptoms, it means that your model is not good. And this is the first thing that is very, very important that we have to respect for the validation of an animal model. The second one is the construct validity. The model must also show mechanisms and causes identical to those observed in humans, the same perturbations. If it's a human pathology or a brain pathology, we need to have the same and identical mechanisms in the dysfunctions of the brain. And the third one is the predictive validity. The model must respond in the same way as humans to different treatments, both positively and negatively. And these 3 points are very, very important for the use of animal models if you want to study the pathophysiology and open the way to a new treatment for a human pathology. And if we classify now the animal models, we have 3 different classifications. The first one is the spontaneous models. It means that in animals, the diseases that occur naturally in them and are identical to human diseases. We have some examples, like for Alzheimer's disease. We know that dogs and dromedary can develop spontaneous Alzheimer's disease. And we have another pathology, which is epilepsy with the Geyer's, sorry, it's not a D, it's a G, a Geyer's rat model, which is the absence, petit mal, absence seizures in rats developed in Strasbourg. The second type of animal models are the induced experimental models with an injury which necessitate surgery, or with nerve ligation when we are working on pain, for example. And there is 2 examples here. Parkinson's disease, we can have, we can induce Parkinson's disease in the rat, mouse, monkeys, and also we can induce in mice the ADHD, which is the hyperactivity of children. And the third type of animal models are the genetically modified models. We can insert a gene to have a model, or we can replace or neutralize certain genes. And these are the three very important types of animal models that we can have. To develop an animal model, we need a sufficient knowledge of the human pathology. If we don't know very well the pathology, we cannot mimic it. And this is why we need sufficient knowledge about it. And we need to know all the symptoms developed in this pathology. And we have to know for a brain pathology what type of brain lesion we have in human to mimic exactly the same lesion in the brains of the animals. And these three points are very, very important also. And if we take today the example of Parkinson's disease, which was discovered or described by James Parkinson, who was a medical doctor in England, he described the pathology in the 1817 in a book with entitled An Essay on the Shaken Palsy. And he described very well all the symptoms developed in human patients, which are akinesia, the invalidity of the patient to do voluntary movements, rigidity, all the muscles of the organisms are rigid with a very high level of the muscle tone, tremor, especially at rest, the perturbation in the posture, freezing symptoms, and gait problems. And all these symptoms were described very, very well by James Parkinson. And this pathology is a neurodegenerative disease because they identify a lesion in the mesencephalon, especially in the pars compacta of the substantia nigra. If we cut the brain at this level, we can see here the location of the mesencephalon in a brain. And at right, you have this part of the mesencephalon with substantia nigra here, bilateral substantia nigra, which is normal from the brain of a normal subject. And if we look to the same region in a Parkinsonian patient, you can see that there is no dopaminergic cells in the pars compacta of substantia nigra. And you can see here the location, the exact location of the mesencephalon with the substantia nigra. And in the physiological conditions, in a normal situation, the substantia nigra dopamine cells project to the striatum in where dopamine is released. And in Parkinsonian patient, as you will have the lesion of the dopaminergic cells in the substantia nigra, there is a cell loss or fiber loss projecting to the striatum, which results in a very decrease, a significant decrease in the concentration of dopamine in the striatum. And with the PET scan using the fluorodopa, you can see here in a normal subject, here in red and yellow, you have the presence of dopamine. And if we look to the striatum of a Parkinsonian patient, you will see that you have only few amount of dopamine inside the striatum. And this is a good demonstration to see the dopamine depletion in the striatum. And the first thing what was decided when the researchers discovered the absence of dopamine in Parkinsonian patients, they were thinking about the replacement of dopamine by dopamine itself. But the problem is dopamine is not crossing the blood-brain barrier. This is why they were thinking about using a precursor. And we know that dopamine is synthesized from the amino acid tyrosine. Tyrosine is transformed with the tyrosine hydroxylase to DOPA, and DOPA is carboxylated to dopamine. Then, with the vesicles, dopamine can be released in the synapse. Here you can see. and can be fixed on dopamine receptors in the postsynaptic level. And you can see that we have two families of receptors, D1 family and the D2 family. And the D1 is composed by two subfamilies, D1 and D5, and the D2 family is composed by three sub receptors, which are the D2, D3, and D4. And the D2 family is coupled with the GI protein, because when dopamine is fixed on D2 family receptors, it will inhibit the cell. We will have a decrease in the neuronal activity. And when dopamine is fixed on D1 family, as it is coupled to GS receptor protein, sorry, the dopamine will exert an excitatory effect. And for that, the researchers were thinking about replacing dopamine by the dopamine precursor, which is levodopa, here, as you can see here. And inside the brain, L-Dopa will be converted to dopamine and will exert its effect on the receptors. And the second way was to develop dopamine agonists to act directly on the receptors. And these are the two ways to treat Parkinsonian patients. The first one is levodopa, and the second one is dopamine agonist. In the beginning, we had only levodopa, but the problem of levodopa is that it functions very well and it is very, very efficient to improve the motor symptoms during the first years, 4 to 6 years. But then the patients start to lose the efficacy of L-Dopa. This is why researchers develop a dopamine agonist to replace L-Dopa in the beginning of the treatment, and after, when it is not, when dopamine agonists start to lose their efficacy, we replace them by the L-Dopa, or we combine the treatment of dopamine agonist with the L-Dopa, and it works very well. And it is very important to start with dopamine agonist to postpone the side effects induced by a levodopa. But at the end, 7 or 8 years after the beginning of the treatment, the patients start to have problems with the side effect of this treatment. And if we look now to the short term, as I said, during the 3 or 4 years after the manifestation of the symptoms, in the beginning, we have a very good effectiveness of L-Dopa or dopamine agonist on the symptoms. We can improve akinesia, we can improve rigidity and the tremor at rest. But in the long term, we have many, many complications who start to appear in the patients. And from these complications, we have the fluctuations of the therapeutical positive effect. The patients can be improved, and a few seconds or minutes he is not improved, and he is not comfortable in his daily life. And the second problems with the dopaminergic treatment is the manifestation of dyskinesia, which are manifested by involuntary abnormal movements. And these are very, very problematic for the patients, because the patients start to move a lot without controlling his involuntary movement. And here I can show you a patient. Here, a patient without any treatment. He is an akineto-rigid patient without a rest tremor. You can see he has very, very low amplitude of tremor, and he is moving very, very slowly. He cannot do quick movements, and this is the difficulty of patients. Now we ask him to do movement with his fingers, but you can see that he has a lot of difficulty to move. And at the right, the patients enter the treatment with levodopa, and you can see that in the beginning, 30 to 40 minutes after taking L-Dopa, he starts to move. These are the involuntary movements, the dyskinesia, and you can see that with time, the amplitude of his movement will start to be amplified. And now at the right, without the treatment at left, he has a very bad balance of his body, and you can see that he has a lot of difficulty to move and to walk. And that's right, you can see that he has also even with the involuntary movement, he has also the difficulties to do precise movements with his fingers and with his hands. And you see that he cannot control these abnormal movements. It's very, very difficult for him to control. And these patients, at this stage, they prefer to stop taking levodopa or dopamine agonists. It's very, very difficult for them to stay all the time like this, moving without controlling their movements. And facing these limitations of dopa therapy, we need to search new therapeutical approaches. And for that, we need to understand the pathophysiology of Parkinson's disease. This is that I told in the beginning in the principles of understanding the pathophysiology. We need animal models because we cannot work directly on the patients. And for that, we need to develop animal models specific to each pathology or brain disease. And for Parkinson's disease, we are very, very lucky, and we have the chance to have many models in rodents and in the non-human primates that were developed in 70s and the 80s. The first rodent model are the 6-hydroxydopamine rat model, or 6-hydroxydopamine or MPTP mouse model, and also we can have an alpha-synuclein rat or mouse model with alpha-synuclein. The advantage of alpha-synuclein is that we develop a progressive pathology in animals that mimic better the progression of the severity of the disease in similar to patients. But the problem is we prefer use 6-hydroxydopamine because it mimics the severe stages of the disease where we have the problems with the patient. And you can use these models with 6-hydroxydopamine or MPTP to search medical, pharmacological, or surgical treatment for the stages with difficulties. And alpha-synuclein models are used especially to develop neuroprotective pharmacological ways. And the second animal model, which is really very good to use because it mimics the motricity of the patients, is the non-human MPTP primate model. We can obtain it in macaques and also in the African green monkeys. And to obtain the rat model of Parkinson's disease, we inject the 6-hydroxydopamine directly in the substantia nigra or in the medial forebrain bundle to lesion the cell bodies in the substantia nigra and also the dopamine terminals in the striatum, as you can see here. We can do it unilaterally by injecting in only one side, or we can use it also bilaterally by injecting in the two sides. And for this, we need stereotaxy to identify exactly the parameters that we can use for the stereotaxy to target with precision the substantia nigra or the medial forebrain bundle. And after finishing the experiment, we can control our lesion if we obtain a unilateral lesion, if we have a partial lesion or a total lesion. And here is a very good example of a total lesion of the substantia nigra. As you can see here, substantia nigra is here. This is an image of tyrosine hydroxylase immunohistochemistry. We can see that in the lesion side, where we injected the 6-hydroxydopamine, we have practically a total lesion of the cell bodies, dopaminergic cell bodies in the substantia nigra compacta. Here it's in the anterior part of the substantia nigra, here the intermediate part, and here is the posterior part. We need to do all the levels to see if we have a total lesion in the whole structure or not. And if we look to the striatum, which receive the dopaminergic projection from the substantia nigra, you can see here the normal side with the tyrosine hydroxylase immunostaining, and you can see that we have a normal striatum. But in the other side, which received the 6-hydroxydopamine, we can see that we have a total lesion of the fibers in the striatum, the anterior part, the intermediate, and also in the posterior part. And here is a magnification to see that we have only few cells surviving after the injection of 6-hydroxydopamine. And this is very good because we need to do this immunohistochemistry to control our lesion. Without this, we cannot validate our behavioral results. And in the MPTP non-human primate model, we can have two models, one with the injection of MPTP intravenously to obtain a bilateral model. And we can see here that with MPTP, we can have a short evolution of the decrease in this binding of the transporters of dopamine in the striatum. And at day 25, you can see that we have a total lesions of the fibers or the transporters of dopamine inside the striatum. And if we look to the substantia nigra, you can see in red the Nissl, sorry, I don't know, my computer is very sensitive. You can see here that in red we have the Nissl staining. No, sorry, we have the tyrosine hydroxylase immunostaining in the substantia nigra, and in blue it's the Nissl staining to see the total cell bodies inside the substantia nigra. And we can see this decrease with time to have a severe, at least 85% of dopamine cells degenerated in the substantia nigra compacta. The problem of this model, the bilateral one, is that the suffering of the animals. The animals are totally akinetic, they cannot move, they have difficulties to eat, etc. This is why we preferred in my team to develop another model, which is a unilateral one that we can obtain by the injection of MPTP inside the intracarotid. When you inject only one time, the MPTP inside the carotid, the internal carotid, you can see that the diffusion of MPTP is limited to only one side, and we will have the Parkinsonian symptoms only on one side, which is the contralateral one of the side of the brain that received MPTP. You can see here with our monkey that we have akinesia and rigidity in his right side, and he is totally normal for the left side. And if you cut the brain of the animals, you can see that we have a normal side with here, you can see the substantia nigra dopaminergic cells stained by tyrosine hydroxylase immunochemistry. And here you have more than 80% of the loss of dopamine cells in the substantia nigra. And this is a very good validation of our model. And then you can use it to study the behaviors, to also understand the pathophysiology in the brain, and also to test molecules for the therapy or surgery for the therapy. And from these animal models, the rat models, and also the MPTP non-human primate models, many studies for more than 30 years research in everywhere in the world, we developed a model of the functional circuits. And we can see we have three parallel circuits in the brain. We have the sensory motor circuit, which is very important for the control of our motricity. If we want to move and to do a voluntary movement, we need these connections to have a control of our acute and especially accurate movement. Here is the second circuit involving the same structures in the brain, but not the same part, because here in the dorsolateral part of each structure is the sensory motor part, but you can see here, these parts are very important for the associative circuit and to control all the cognitive and functions. And at right, these are the ventral part of all the basal ganglia structures, which are involved in the limbic circuit to control emotions and the limbic functions. And these models were very important to develop this kind of model, which helped us to understand the pathophysiology of Parkinson's disease and then try to find a new therapeutical approach to try to improve the symptoms in the patients. And we have a very good example for the things that we developed. And to come back to the Pathophysiology, here it's a kind of modelization of the circuits. And you can see that the cortex, when the cortex gives information to the basal ganglia to control a movement, you have here the entrance of the signal coming from the cortex to the basal ganglia through the striatum. Then the striatum will communicate the informations to the output structures of the basal ganglia via the direct pathway, as you can see it here, which is GABAergic inhibitory, and here an indirect pathway. And the subthalamic nucleus is located at this central part, which is very, very important for the control of movement. And then the output structures send the finalized informations to the thalamus. Then the thalamus sends the information to the cortex, and the cortex sends the final decision to initiate the movement by sending the information to the brainstem and the spinal cord. And this is a normal situation in which, if you look to the structures of the basal ganglia and you do electrophysiology by recording the neuronal activity in the globus pallidus externus, or in the subthalamic nucleus, or in the output structures of the basal ganglia, you can remark that in all these structures, you have a tonic regular activity in the normal situation. We have exactly the same pattern of discharge with electrophysiology in the subthalamic nucleus and also in the external part of the globus pallidus. These are the spikes, the action potentials that we record from each structure. And if we look now to these basal ganglia structures with electrophysiology in animals after the depletion of dopamine, you can see that this tonic activity is replaced by bursty activity. And this bursty activity is a pathological signature of basal ganglia in Parkinson's disease. And we can find this bursty activity in the globus pallidus externus, in the subthalamic nucleus, and also in the output structures of the basal ganglia, the substantia nigra reticulata, or the internal part of the globus pallidus. And these are really the pathological signatures of basal ganglia in Parkinson's disease. And the question was, if we can interact with this bursty activity, is it possible to improve Parkinson's disease? And the first idea was coming from Haggai Bergman in Mahlang Dolong lab in Atlanta in the United States, who was thinking about lesion in the subthalamic nucleus in MPTP monkeys and see if we can improve the symptoms. And the results were very, very important for us because they showed an improvement of the motor symptoms in the MPTP monkeys with the improvement of akinesia, rigidity, and also tremor. But the problem is, as the subthalamic nucleus is known to be involved in hemibalismus, in the manifestation of abnormal movements, because in humans, we know that if you have a lesion in the region of the subthalamic nucleus, patients develop hemibalismus by the manifestation of abnormal movements that they cannot control. And all the monkeys which received the lesion of the subthalamic nucleus developed hemibalismus. And as the lesion is an irreversible approach, it was not possible to propose this lesional approach to Parkinsonian patients. And this is why we were thinking in our team to replace the lesion by an electrical stimulation, and by playing on the parameters of the stimulation, we can control the neuronal activity of the subthalamic nucleus. And we proposed to replace this lesion by high-frequency electrical stimulation to mimic the lesion of the subthalamic nucleus. When you are thinking about the electrical stimulation, we think automatically to an excitation. But in our case, high-frequency stimulation is not exciting the cells, but it inhibits the cells, because the cells in the subthalamic nucleus cannot follow the high frequency that we are imposing on them. High frequency stimulation has an inhibitory effect, and then we can play on the intensity for the fusion of the current inside the nucleus. And with our high frequency electrical stimulation of the subthalamic nucleus, we obtained a dramatic improvement of the motor symptoms, of akinesia, for rigidity, and also for tremors. And the most important thing was also that we got no side effect, no involuntary movements at the intensities used to improve the motor symptoms. But if you increase at a very high level the intensity, you can obtain the involuntary movements in our animals. And as it is a reversible approach, it was able to propose this approach, this neurosurgical approach to the patient. And this is a demonstration of the effect of the lesion, the high frequency stimulation instead of the lesion of the subthalamic nucleus on the tremor. Here you can see the vertical lines are the tremor, expressing the tremor. And when you start to stimulate at 130 hertz, we can see that we stop totally the tremor. And when you stop the stimulation, the tremor is coming back few seconds later. It means that we need to stimulate all the time to stop the tremor. If you stop the stimulation, the patient is coming back to his pathological situation. And if we look to the rigidity, here is, as our animals are unilateral, they have Parkinsonism in only one side. If you record the muscle tone inside the muscle, here, the biceps, for example, in the normal situation, at this level, you can see that we have a low amplitude of muscle tone. But if you record the muscle tone in the biceps of the rigid member, you can see that we have a high level amplitude, which is expressing the rigidity in this part. And when you start to stimulate, here is a superimposition of many traces, 10 traces of EMG recordings. You can see this high level, which is reversed when you start the stimulation of the subthalamic nucleus. We can normalize totally the muscle tone. It means we are normalizing rigidity. And these were a very good demonstration to show that we have an improvement of rigidity and also with the tremor. And we showed, sorry, we showed also that we can play by improving the akinesia in our monkeys. And with these encouraging results, we proposed the transfer of our subthalamic high frequency stimulation to Parkinsonian patients. And this is why I moved from Bordeaux to Grenoble to participate in the transfer of this technology. And we can see here that we implant the electrodes in the subthalamic nucleus exactly in the same way that we are doing in our animal models. Then you connect the electrodes to a stimulator here with a cable. And you can see that this stimulator can control both sides, and we can control all the parameters of the stimulation by a computer. We can adjust the frequency of the stimulation, the pulse width, and also the intensity that is improving the symptoms. And you can see that we have in our patients a dramatic improvement of the cardinal motor symptoms, rigidity, akinesia, tremor, and the posture and gait also problems. And here to see during how we are carrying out the surgery. It's a very good movie that summarize how we implant the electrode inside the brain. Okay, here is the thalamus, here is the subthalamic nucleus that we are targeting. We put a stereotactic frame on the head of the patient. This will help us for the MRI control. We put the human patients in the MRI to have the images. Then we calculate the trajectory on the human slices. And you can see here the subthalamic nucleus on a slice of MRI images. And then we prepare the head of the patient to put the holes, and we put the supporter of the electrodes. And you can see here that the patient, the medical neurosurgeon is preparing the head, and we put the micro electrodes. We are using 5 micro electrodes to target the region of the subthalamic nucleus, and we record the neuronal activity to see this burst of activity. Then, as the patient is not under general anesthesia, he can collaborate with us to see if it is working, the stimulation is working or not during surgery. Then, when we are sure that surgery is functioning and the stimulation is reversing the Parkinsonian symptoms, we replace the microelectrode by the macroelectrode, and then we insert the stimulator under the skin, and we can control the diffusion of the current from the stimulator to the electrodes and to the subthalamic nucleus. And here, if we compare the electrophysiological results that we obtain in our human patients to animal models, you can see this bursty activity exactly the same that we obtain in our animal models. And this is the demonstration that our electrodes are crossing the subthalamic nucleus. And if you look here, I told you that we are using 5 microelectrodes to explore the neuronal activity in the subthalamic nucleus of the 2 regions. You can see that it's very, very small. The subthalamic nucleus is a very small structure. And by using these 5 microelectrodes, we can have at least 2 or 3 microelectrodes inside the subthalamic nucleus. Then we select the one with this bursty activity to replace the microelectrode by the chronic one. Here you can see in patients with tremor that we can have a very good synchronization of the tremor that we can record by accelerometry. Each peak corresponds to a tremor. And here the bursty activity of the subthalamic nucleus. And you can see a very good synchronization between the tremor and the bursts, as you can see here by the recordings. And this demonstrates that the tremor is depends also on the bursty oscillations in the subthalamic nucleus. During surgery, we record the neuronal activity, and you can hear the bursty activity with the tremor of the patient. You can hear the tremor.
Synchronisé avec le tremblement. On va arrêter le tremblement.
This is also a good demonstration. Now there is no burst because the patient is not tremoring.
Et regarde, on a une activation avec le poignet. Regarde.
The responses of the cells to the movement.
On voit bien qu'elle répond au poignet.
We have an increase of the neuronal activity in response to the movements of the patient. And this is very good. It expresses that we have the electrode in the sensory motor part of the subthalamic nucleus. And now we are coming back to the tremor. Yeah. You can hear the bursty activity synchronized to the tremor. Then, when we are sure that our electrodes are inside the subthalamic nucleus, we can test the beneficial effect of the stimulation on the symptoms. And here you can see the patient with his tremor. And we will see when we start the stimulation, we will stop his tremor. Stop. The stimulation started and the patient is not tremoring. We can stop the tremor with the stimulation of the subthalamic nucleus. Then you can see also the tremor, and we will start the stimulation in few seconds. Stop. We stimulate, and you can see the stopping of the tremor. It's really an immediate effect by starting the stimulation. And when you stop the stimulation, the patient starts to have his tremor. And at the end, I told you that we replace the microelectrode with the chronic electrode. In this macro electrode, we have 4 contacts. You can see here, we have 2 contacts inside the subthalamic nucleus bilaterally. And then the patient will stay in his life with these macro electrodes inside the inside the subthalamic nucleus. The duration of life of the stimulator is around 8 years, and the patients come back to the surgery department to replace only the stimulator. We are not touching the micro and the macro electrodes in the brain. Here is an example of a patient with a high level severity of tremor. Here it was before surgery, patient without stimulation. We are asking him to do movements, and you can see that he is perturbed by this tremor, and he cannot move correctly. And we will see with the stimulation, we stop totally his tremor, and he can move normally. We will ask him to do the same movements like before. You can see that we stop tremor in the legs and also in the hands, everywhere. All the body is concerned. And now he can do the movement that he wants to do. And look to the exact finalization and of the movement that he is executing. It's really very, very important. Coming back now before the stimulation, without stimulation, we are asking him to do voluntary movements to open his hand. And you can see that he has a lot of difficulty with to open his hands quickly with this perturbation with his tremor. You can see here that he is trying to do precise movements, but he is not able to do this precise movement. And after the stimulation, you can see that he is doing normal movements without any problem. You can see the precision of his movement of the fingers that was impossible to do before surgery. Now we are asking him to stand up, and you can see that he has a lot of difficulty to stand up, and he's trying to block his tremor. But now you can see his tremor. He cannot turn very easily. He's doing it by steps. And after the stimulation, we are asking him to do the same movements, and you can see that he stands up very, very normally, and he can move without any problem. Then we ask him to walk, and you can see the walk of a Parkinsonian patient with very short steps. And after the stimulation, you can see he is walking totally normally. If you look to all the parameters of the movements, we are normalizing these patients at all the levels. And here is another example, a young woman. Now she is Parkinsonian, but she is on stimulation.
What that looks like. So I've got arms that can stay still instead of shaking. Hands that can do this. I can put my arms up, and I can lift my leg up.
She is moving totally normally because she is stimulated, and you will see when she will stop the stimulation.
To show you what it looks like. So this is my
yeah, she can stop the stimulation with this machine.
It's synchronized to my battery, so you can see that it is on and okay.
Yeah, the stimulation is on.
And my DBS is on. If I push this white button, it will turn it off. So here goes nothing. Disclaimer, if stage 4 Parkinson's bothers you, don't keep watching. I'm off.
Yeah, you can see it's immediate. She came back to her Parkinsonian symptoms.
In my shoulders.
Yeah, and you will see the aggravation with time.
You can hear the change in my voice.
Yeah.
My throat muscles are having to work a little extra hard. I can't lift my leg up.
Yeah, she becomes a Parkinsonian after stopping the stimulation.
I'm going to turn it back on.
Yeah. She is not happy to do this, and she will retry to start on the stimulator.
It's just my facial muscles failing. So here we go. Very good.
Yeah.
It's literally as simple as that. It's back on now.
Yeah.
That's DBS. That's why I'm doing surgery.
And what is amazing is that immediate effect when we start the stimulation or when we stop the stimulation. It's really immediate effect. Well, and I showed you two perfect patients that we are improving very, very goodly. But all the patients are not improved in the same way. And we try to understand why some patients are not improved in the same way. We have only partial beneficial effect. And for that, we try to look to the systems that in the brain that are involved in the motor control. And we were thinking about the monoaminergic systems, because the Parkinson's disease is not a pure dopaminergic pathology. We have also the noradrenergic and the serotonergic systems which are affected in Parkinson's disease. And that we try to do is to develop a stimulator implantable in the rat and try to interact with the monoaminergic systems, with the noradrenergic and serotonergic systems, to see if the improvements are also obtained in animals with additional lesions of the noradrenergic and also the serotonergic systems. And you can see here our stimulator with the animal at the end with his stimulator. Here are the holes that we prepare to put the electrodes bilaterally in the subthalamic nucleus. This stimulator, we developed it, it was developed by Houyam Tibar, who is a neurologist in the Rabat University, and my team, other members of my team. Houyam Tibar did her PhD with me in Bordeaux, and she worked with Frédéric Naudet and other people, Emilie Fagiani, who were involved in this work. And we can see here, by lesioning the noradrenergic system and the serotonergic system combined to the dopamine lesion, or the three combined lesions of the three monoaminergic system, you can see that we have locomotor deficiency in all our animals without any potentialization of the locomotor activity. The hypolocomotor activity is obtained with all the lesions. There is no aggravation by additional lesions of the other systems. But here, what is important is to see that we are improving locomotor activity during the brain stimulation. Here in red is with DBS, and in blue without DBS. And we can see that with DBS, we are improving in the dopamine depleted animals, we are improving the locomotor hypoactivity. However, if you add a lesion of the serotonergic system to the dopaminergic lesion, you can see that we are altering the beneficial effect of the brain stimulation. We are not improving anymore the locomotor activity in our rats. And you can see that we have exactly the same result when you combine the noradrenergic lesion with the dopaminergic lesion. And here also with the three lesions, we have no significant improvement of the hypolocomotor activity. And we have exactly the same result with anxiety. You can see here that deep brain stimulation is improving anxiety in the rats with only dopamine lesion. However, if you have the additional lesion of noradrenaline or serotonin, you are altering this beneficial effect on anxiety. And here you can see also the same problem that we have. It means that in the patients where the subthalamic nucleus stimulation is not working very well, we think that in this patient, they don't have only a dopaminergic lesion, but they have additional lesions in this monoaminergic system. Now we are thinking about a project, in a clinical project with the PET scan, to see what happened in our patients who are not improved very well with the stimulation, to look to their serotonergic and noradrenergic systems if they are affected or not. And in conclusion of my talk, I showed you that subthalamic high-frequency stimulation is a therapy of choice in the current state of Parkinson's disease treatment. We obtain a very, very stable beneficial effect with time. And this is very important because it's not the case with the DOPA therapy, but by stimulation of the subthalamic nucleus, we have a stable effect with time. during the stimulation of the subthalamic nucleus. And this, the beneficial effect of subthalamic nucleus can interact with the noradrenergic and serotonergic systems. And in the case of Parkinson's disease research, I clearly did demonstrate to you the key role of animal models in biomedical research. We can have this example to say that we cannot work without animals to find new approaches for human diseases. And the use of animal models is very important in the understanding on the pathophysiology of human disease, and without which no treatment can be developed. And to finish, I would like just to show you during my PhD, I was supervised by Bernard Bioulac and Christian Gross during my PhD, during which we developed high-frequency stimulation of the subthalamic nucleus in the monkeys. Then I moved to Grenoble to reach the team of Alim-Louis Benabid and Pierre Paulac, where we did the first patients in the world with the brain stimulation. And here are people working in my team. This is the Neurochemistry, the brain stimulation and Parkinson team in the Institute of Neurodegenerative Diseases in the Bordeaux University. Thank you very much for your attention.
Thank you so much, Dr. Abdelhamid. It's been an amazing talk. I think one of the joy one derives from research is if you can really see the product of your work really being translated already. Many don't get to see what you've seen already. So many, maybe years after the discovery, I think I was excited when it got to the high frequency stimulation part. So I will read out some of the questions we have because we don't have much time. And then I will also ask my question afterwards. And then you'll have to advise us at the end on how to have an amazing career in science, hopefully. So the first question here, I think the question was early on in the presentation, and I think you've already answered it, but maybe you just mentioned it again. The person says, what type of animals, mice or rats, can be used in PD research? What is the best animal model or animal for PD, no, best animal for PD research? I think you answered this, but just to mention it again.
Yeah, in my experience, in the beginning of my career, when I was PhD student, I started directly to work with the monkeys. Why I started to work directly on the monkeys? Because many works were done in rodents to understand the pathophysiology. And at the time when I started my PhD, we had many ways to think that if we target the subthalamic nucleus, we know that the subthalamic nucleus is problematic in Parkinson's disease. And the animal which is the nearest with his motricity to human is the monkey. The monkey has the same motricity, has the same motor circuits, etc. in his brain. And this is why we started directly to work with the monkey. And when we validated the results on the monkeys, it was possible to transfer this technology to human. But when we don't know a lot of things about the pathophysiology, we have to start with the rat. And when we obtain encouraging results in the rat, you can move to the monkey before proposing something to human patients.
Yeah.
For people, if we are working now, actually, if we are working on the neuroprotective ways to avoid Parkinson's disease, the best way is to start with animals, with rodents, and not the monkey. I think somebody who has a microphone open. Okay, if you start to work on something which is not very clear, it's better to use rodents. 6-hydroxydopamine rat is the best model in rodents to explore the late stage of Parkinson's disease. And MPTP model is the gold standard model of the disease. You can use it when you are sure that you have promising results from rodents. It depends on the question of your science.
So the second question we have here, I think it's kind of general. The person says, are there data about the effect of specific nutrients on PD treatment or diagnosis? So the person says, are there like specific nutrients or maybe the food that one can take that can help ameliorate the symptoms of PD?
Ooh, this is a big question. The problem is we cannot treat Parkinson's disease without medication and without surgery. The nutrition is an additional help to treat the patients, but you cannot treat Parkinson's disease with plants or with foods. It's sure that it cannot work. And that we know now is for a good efficacy of levodopa treatment in patients in the beginning of the disease is to avoid eating proteins at the time when the patient takes his medication, because there is an interaction for the absorption of the levodopa in the body and in the brain. This is why the patient has to avoid to use proteins in the time of eating, in the time when he is taking his treatment with levodopa.
The next question we have here, it's like a career question, so we'll come back to it at the end. So, but another question says, is the surgery safe? What are the most common complications for electrodes use?
Yeah, never somebody can say that surgery is safe, because surgery is invasive and you will put something in the brain. We are putting electrodes in the brain. The advantage that we have now compared to the 90s when we started to work with this kind of surgery is that we have now MRI controls that we can see on the images, all the vessels, and we are selecting the trajectories where we are sure that we will not cross the vessels. And this improved a lot our problems of side effects with surgery, because before we had always the risk to have hemorrhage in some patients during surgery. But now we don't have this problem because of the evolution of the technology. Now the MRI control with gadolinium, we can see exactly the positions of the vessels that we can avoid for the trajectories that we are selecting.
So my own question is this. For the patients that you do the high frequency stimulation for, are these patients that had already been on medication on maybe the dopamine agonist or the L-Dopa, and now they have tremor from the adverse effect, or because the drugs are no longer working? Are they the ones that you do the high stimulation for? Or people come in at even early stages of the disease?
Yeah. In our case, and as in the majority of centers in the world who are doing surgery for Parkinson's disease, we don't start with deep brain stimulation in patients when they started the disease. We prefer to start with medication, and we operate them for the implantation of the electrode at the time when we have no efficacy with the treatment. Because as medication is working very well during the first years of the disease, we don't want to give them the risk of surgery. We prefer to treat them by medication, but when they are losing the beneficial effect of the medication, we propose surgery. And at the same time, we cannot operate all the patients with Parkinson's disease because we have now very good list of criteria of inclusion and exclusion. And I can give you an important example. The first very good example is depression. We know now that in majority of patients, they start to develop depression before the movement problems, the motor problems. And in patients where we have a very severe depression, we prefer to don't operate them because we know that subthalamic high frequency stimulation will aggravate their depression. We have some example of suicide, for example. And this is why we are doing a very big job and a very big work with neurologists and psychiatrists to be sure that the patients that we want to operate are not depressive severely, because we know that we will aggravate his depression. And we have other symptoms, especially psychiatric symptoms, because Parkinson's disease is not a pure motor disease. We have also some psychiatric troubles in Parkinsonian patients. And in the patients with this kind of psychiatric problems, we prefer to don't treat them and try to adjust the dosage of levodopa combined with dopamine agonist or apomorphine, etc., to have a stable effect on their motor symptoms. And surgery is proposed only to people with Parkinson's disease without severe depression.
Thank you so much. So going to the last question, which is like very general. A person asked, what type of research do you suggest them to do in Parkinson's disease? But I'm going to combine that with the general question. So yeah, most of those listening are early career researchers. So yeah, while answering the question, I would also like you to kind of give a career advice to everyone listening.
Yeah, if I understand well, yes, what is the way that we have to choose now to do research on Parkinson's disease?
Yes, that's the impression I have too.
Okay.
Yeah.
For me, the way that we can have now, which is very, very important, is the neuroprotective way, because we are trying now to interact with the symptoms. We know exactly how we can do to improve the motor symptoms, who are the people who can benefit from these approaches that we have now, etc. But the best way will be to avoid the development of Parkinson's disease. And to avoid to develop Parkinson's disease, we need molecules who will interact with cell death. This is why I'm talking about the preventive procedures or preventative approaches to avoid the cell death. And this is very, very difficult, but this is the major way for all the neurodegenerative diseases, not only for Parkinson's disease. And many labs are working on these ways, but it is very, very difficult for the moment to find a solution to avoid cell death. Many molecules are working very well on animals, and all the clinical trials to test these molecules was aborted because it was not the best molecule to use. But I think the best way is to continue to discover other molecules to interact with the cell death and try to stop the evolution of the disease, or one day perhaps we will stop cell death in general.
So, and then the last part is your advice for the early career researchers that are listening to your talk now on how to hopefully have an amazing career.
Science. It's a very big question, you know. A very big career is to find something in research which serve human. And I think it's not easy to work on this, but we have to stay positive and try to do the best to improve our quality of life and to fight the pathologies, human pathologies. You have to be motivated, work hard. You cannot be successful in your career if you are counting our time in the work. I think for science, you have to work very hard. You have to try to develop new things, not to just reproduce that the others did. And this is, I think, the points that are important in the career of a researcher. You have to be ambitious, to try to think about new ways to develop new technologies and to work on the fields where a lot of questions are still here. And I think if we are positive, if we are ambitious, if we are motivated, if we are not counting our time, I think we can be successful in our career.
Thank you so much. Before I let you go, I'm sorry for taking your time. We have this important question that just came in. So when you were mentioning the models for Parkinson's disease, there's a common model, the Rotenon model. So the person is asking if this is still relevant for as a model for Parkinson's disease.
The Rotenon model, I don't like to speak about it because I didn't present it in my list of models of Parkinson's disease because it is very difficult to obtain. We cannot obtain it in all the species. There is only few rats that can develop Parkinsonian symptoms with Rotenon. Rotenon is not, for me, is not a good molecule or intoxicant to use to reproduce in all the species Parkinson's disease. It's really very, very difficult to obtain. This is why I prefer to use something which is sure inducing Parkinson's disease. Instead of injecting Rotenon, inject 6-hydroxydopamine, we will obtain a more reproducible model of the human Parkinson's disease. For me, Rotenon is not the good model for Parkinson's disease.
So we have a lot of people thanking you for being a good model to them. So, and before we say thank you again, I would like to congratulate you and everyone listening for doing something that is benefiting humans directly. I think, like I mentioned initially, not everyone has the privilege of really seeing their work go translational. So on behalf of SONA, the IPDGC Trend in Africa, we want to thank you for honoring our call and taking that time out to educate us all. And we hope that in the future, if we really want to listen to more of this, which I'm sure we will, you will be glad to honor our call.
It will be with pleasure. I really thank you very much to organize this kind of webinars and conferences, inviting people from everywhere from the world. And I think this is very, very important for the development of research in Africa. I like Africa, I'm African, and I will do all my best to help Africa. Thank you very much again. And
thank you so much. Thank you so much, Dr. Abdelhamid.
Thank you.
Goodbye. Thank you so much. Bye.